All right, it's now the top of the hour. It's 7 p.m. Eastern, so we're going to go ahead and get started. Good evening and welcome to tonight's webinar. This evening, we are joined by Dr. Joud Hajjar, who will share an overview of her study on PI and the microbiome. My name is Emma Mertens, and I'm the Program Manager of Community Health at the Immune Deficiency Foundation. On behalf of the Foundation, we thank you for tuning in. Tonight's webinar is one of many virtual education opportunities available in our 2023 programmatic lineup. We recognize that through the provision of timely, comprehensive, and accessible information, we can improve the diagnosis, treatment, and quality of life of people affected by primary immunodeficiency. We appreciate you being here as part of this journey. Before we get started, I would like to point out a few tech pointers to keep in mind for tonight's program. This evening, we are using the Zoom webinar feature, and attendees should be able to see the slides and hear the presenter and host speak. Attendees will not be able to activate their video camera or their microphone. There will be the opportunity for questions after the presentation. You are welcome to submit any questions you have for Dr. Hajjar as you think of them throughout the session. We kindly ask that questions are relevant to tonight's webinar topic. Please type them once in the Q&A box in the control panel on your screen. Please do not include any personal health information, as all questions will be anonymous and read aloud. A brief disclaimer. Please remember, the information presented during this meeting is not intended to be a substitute for medical advice, diagnosis, or treatment. We are here today as a trusted source and friend to provide you with information. Always seek the advice of your physician or other qualified health provider with questions concerning a medical condition. Never disregard professional medical advice or delay seeking it based on information presented during an educational event. Be sure to check out our website at www.primaryimmune.org, where you'll find resources, events, support services, and more. Everything is free to access and print. We can also help you find an immunologist or PI specialist in your area using the Clinician Finder tool. IDF hosts educational programming and events, including lunch and learns and webinars, which cover a variety of topics facing the PI community. We also offer Get Connected groups, peer support, Ask IDF, and our annual PI conference. Are you living with PI, or are you a parent or caregiver to someone living with PI? If so, we encourage you to take IDF's health insurance survey. These surveys have helped to document the financial costs associated with seeing providers and overall health insurance experiences. The results have been used to better advocate for the commuter to policies and payers alike. It's now time to update this important information with your experiences. Your honest responses are vital to the research and will help guide IDF efforts in improving healthcare delivery for our community. To participate in the survey, please scan the QR code on this slide. This meeting is made possible by our wonderful sponsors. It is due to their partnerships and contributions that we can provide programs like this for the PI community. Please join me in thanking our 2023 sponsors: CSL Behring, Grifols, Takeda, Octapharma, Pharming Healthcare, Horizon Therapeutics, AstraZeneca, ADMA Biologics, Sumitomo Pharma America, X4 Pharmaceuticals, CVS Specialty Pharmacy, and Chiesi Global Rare Diseases. Thank you all so much. And now I am so pleased to introduce our presenter for this evening. Dr. Joud Hajjar is an associate professor of immunology, allergy, and rheumatology at Baylor College of Medicine. She is also the service chief of adult allergy and immunology at Texas Children's Hospital. Hajjar is a longstanding member of IDF's Medical Advisory Committee and a 2022 IDF Research Grant recipient. Thank you for joining us, Dr. Hajjar.
It's my pleasure to be here. Thank you for having me. And I'm just gonna take a second to share my slides. Everything looking good from your end?
Yep, looks great. Yep.
Wonderful. Well, good afternoon or good evening, everybody, depending on where you are. I'm so grateful to be with you this afternoon. I'm so excited to share with you some of the work that we've done, but more importantly, kind of try to talk a little bit more in depth about the microbiome and its role in primary immunodeficiency disorders. Here are my disclosures. I do receive some consultation fees and some honorarium, but none of the work that I'm going to be presenting today or the data that I will be providing would be of conflict of interest, with the exception that our human-based work has been supported by the Immune Deficiency Foundation. You know, talking about the microbiome and immunodeficiency is a really large topic. So what I was hoping to do today is to bring you all the way down from the basics of the microbiome. What is really the microbiome? And how is the microbiome developed in humans? When does it develop? And what are the microbes that shape the microbiome? And we're going to touch upon very briefly and in a very basic way about some of the research that has been done in immunodeficiency disorders to kind of try to understand the interplay between the immune system and the microbiome. And we're going to use the healthy individuals who are immunocompetent first to try to understand how the immune system and the microbiome should interact in an immunocompetent individual. And then we're going to contrast this to individuals who have immunodeficiency. And for that, I'm going to use the example of the disease common variable immunodeficiency for multiple reasons that I will touch upon during this presentation. And I tried to leave a good part of this presentation to talk about microbiome-based therapeutic approaches. What do we know about them so far? Are they helpful? And what can we learn from them? So let's start by really going to the basics. What is the microbiome? So this term microbiome was coined by Dr. Joshua Lederberg. And when he coined the term microbiome, he was referring to an ecosystem of friendly, neutral, and sometimes harmful microorganisms that live inside us. So very early on, providers and scientists have understood that there are other microorganisms that live inside the human beings, and they understood from the get-go that some of them are beneficial, some of them are neutral, but some of them can be harmful. Now, when we're talking about the microbiome in scientific journals or in scientific meetings, we're really referring to the collection of genetic material from all the microorganisms in specific place. So, for example, when I'm talking about the gut microbiome, which is the topic that I will be discussing today, I'm referring to the genetic material from all the microorganisms that are there. The term microbiota that may come across in some of the papers that you read on the internet refers to the actual microorganisms like the bacteria, the fungi, the viruses, and so on. And the reason we do this discrimination is because all the ability for us to identify bacteria is by doing genetic sequencing on those microbes, whether it's bacteria, virus, or fungi. And that's why those terms are not truly interchangeable. So, what are the physical properties of this gut microbiome? The physical properties are very important because there is a benefit of the mere physical presence of the microbiome in our gut. It weighs between 4 to 5 pounds in a healthy adult individual. And if we take all the body microbiome, we now know that about 40% of all body microbiome exists in the gut. And as you can imagine, this will lead to a large surface area of interaction between the gut and between the immune system. And that's why we pay a lot of attention to the microbiome as clinical immunologists. So what does this microbiome do? As I mentioned earlier, the mere physical presence is very important because if you have microbiome that is enriched by friendly and health-promoting bacteria, that will just occupy the space, allowing less and less bad players, or what we call pathogens sometimes, to be present in our gut and in other parts of our body as well. As we're going to see, the microbiome interacts significantly with the immune system, and it will impact the immune system, but it also plays a major role in immune system maturation. And we know a lot of this from mouse studies, in which mice that are living in what we call a germ-free environment really do not have a mature immune system. And in some ways, they look like CVID patients because they have low immunoglobulins, small lymph nodes. And that's because they have not seen any microbes and they have never been infected. They even did not encounter them because they live in incubators. Once you take them out and you just put them in a regular environment, their immunoglobulins go up and their lymph nodes become normal in size and their immune responses improve, telling us that the microbiome really plays a major role in maturation of the immune system. And those microbes are not there just for the physical presence. They are really metabolic. They are really metabolic machines, and we consider the microbiome to be a metabolic organ. Those bacteria metabolize the nutrients that we ingest and play a major role in shaping our immune system through those metabolites, which is part of the work that we're working on, and I'll touch upon briefly about it. The last part that I will not have time to discuss today is the impact of the microbiome in behavior and mood. There are a lot of studies right now that talk about the gut-brain axis and the impact of the gut microbiome in mental health and on specific mental disorders. And this is really a large area of research right now. The gut microbiome in humans develop right at the birth, whether the baby is being born through vaginal delivery or through cesarean section. The baby is introduced to bacteria, as we believe, for the first time as they go through the birth canal or they are delivered. And as this picture shows you here, the babies, when they are born, they really have very small amount of microbes with what we call very minimal diversity, meaning, as you see here, there are only a couple of microbes that are present in the stool of a newborn. But this increases significantly even within the first couple of months. It doubles, and as the babies start to move around, put their hands in their mouth, start consuming solid food and so on, and they get exposed, their microbiome gets enriched, and there are way more and I apologize that those slides are advancing on their own. Those microbes get enriched significantly, and this increases towards the first few years of life. And you really, after the microbiome gets enriched, this microbiome, we expect for the most part to become stable over time. There are a lot of factors that could shape the microbiome development very early on in life. And there are multiple factors. Some of them play a major role. As you see here, some of the genetics, some of those factors are dependent on maternal factors during pregnancy, exposure to medications, exposure to antibiotics, the term of delivery itself, the methods of feeding, and when the baby was weaned, and whether they were breastfed versus bottle fed, and whether they were exposed to antibiotics early in life. There are other factors that play a role, but maybe less impactful, like whether there are pets in the house, whether the babies were exposed to certain foods, how many siblings they have, which part of the world they live in. All of those factors impact that very early development of the microbiome, which is significantly important. Now, there are a couple of concepts that I think are very important for all of us to understand about the microbiome responses, which will help us understand why there are big differences between people who are immunodeficient and people who do not have an immunodeficiency. And for that, I would like to use the analogy of a garden. So, imagine that you have this beautiful garden, that you have different kind of plants and roses and flowers, and you have different colors and different types and different shapes. And this is what we call a diverse garden. And this is what we refer to as a diverse microbiome. So, all of us will undergo changes in our life, in our environment, in our diet, but a healthy, the microbiome, if it encounters a short and not very impactful change, it will be resistant. So let's say your beautiful garden, there was a short storm, it rains, everything will be okay after the rain goes away. So you're not going to lose any flowers normally if there's just a short rain. And all types of flowers and plants that you have are the same. And the same thing here. If the microbiome encounters a short-term change or disruption, like somebody changed their diet for a couple of days because they went on a trip and they ate different cuisines and things like that, if you are starting from a strong microbial community, it will stay stable and there will be no change. However, there are times in which the impact is a little bit stronger. So let's say that there was a big storm with freeze or a hurricane or several days of rain that did not stop. Maybe some of the plants will die down a little bit. Maybe some of the flowers will die for a few days. But when everything is over, maybe those flowers will just regrow and those plants regrow. And this is what we refer to in the microbiome world as resilience. So if there is a temporary issue, like there was an infection, somebody got on a course of antibiotics, there was a diet change for a longer period of time, maybe the microbiome composition will change. So maybe like some of your flowers will disappear, maybe some weed will show up also. But all in all, after all those problems go away, your garden will go back to normal. And again, your microbiome will go back to the exact same state that it was before all those problems happened. And this is what we call resilience, which means we went back to exactly where we started after a period of recovery. The third level of response to change is the adaptation, which means that you're adapting to long-term change. So let's say there was a significant change in the environment. Now there is a drought and you cannot water your backyard as regularly as you can. Maybe the soil supplier has changed. Maybe your backyard has become smaller, so you cannot plant all the garden flowers and plants that you had before. But you will go ahead and plant other things that will give you a mutually beautiful garden, but it's not exactly the same. It's different. And this is the same with the microbiome. So maybe somebody travels across continents and they change diets completely. Maybe somebody moves from being an animal meat eater into complete vegan. And those changes are going to be permanent, or they will take a very long term. The microbiome is going to have to adapt to this, and maybe it will change its composition. The end result may not be exactly the same microbiome composition like you had before, but it will be mutually beneficial. So you're not going to have your same garden that you started with, but you're going to have an equally beautiful garden, but with different flowers, but it will give you the same joy. And for the microbiome, it will give the same function to the host. Now, the last example that I'm going to give is the problem, is when the microbiome fails resilience. So when there is a huge change and you're starting not with a very strong microbial community and a big change happens, then it cannot bounce back. So if your garden did not start healthy and now there's no watering, the weeds are growing, the flowers died, and they are just not taken care of, they're not going to be able to go back again to be a beautiful garden. And maybe now there are some flowers that are growing, but there are more weed that is growing, and all in all, it is not a healthy garden. And same with the microbiome. Sometimes if the change is so severe and the microbiome is starting not with a strong community, now the end result microbiome community is not very helpful to the host. And this is what we refer to as resilience failure. So what determines whether people can have resilience or not? There are factors that are dependent on the microbiome composition itself. There are factors that are dependent on our genetics, and there are factors that are dependent on our immune system. So if you're starting from a very diverse microbiome, that's an indication of strong microbial community, because even if the antibiotics came and killed part of your good bacteria, if you have a very diverse microbiome, there will be a lot of redundancy in the function of those microbes. So some die, but there will still be some that are able to continue to carry on and function. Just like your garden, if you have a wide variety of flowers and roses and plants, and a storm happens and some of them cannot adapt to the bad weather, some of them might become more resilient and will still be able to make this garden look good. There are host factors. So there are some genes that are play a major role in shaping the resilience. And you might have heard about this gene, of some of you who had genetic testing done, you might have heard about the NOD2 gene. And some variants, which we used to refer to as mutations, make that gene do not function very well and cause increased risk of susceptibility to inflammatory bowel disease. And this is directly linked to some changes in the microbiome, making the microbiome less resilient. The third part is that talk between the microbiome and the host, and this is significantly mediated by the immune system, as I'm going to show you in the next slide. So this is a cartoon suggesting that this is the lumen of the gut, and those are the epithelial cells in the gut, and this is the lumen where usually the bacteria and the stool will exist, and towards the inside is we're taking a cut through all the layers of the tissue, and those just magnified plasma cells that make the antibodies. And as you would expect, there will be a lot of bacteria here. Some of them are friendly, some of them are neutral, and some of them are trying to be bad. But in general, our immune system, particularly our antibodies, very specifically IgA and sometimes IgM, are like a wall here that come in and bind to those bacteria, particularly the ones that are bad players, and does not even let it in. So they cannot go in and they cannot do anything. They just get eliminated. And IgA does this in a very quiet way. It's like a wall. You cannot pass through it. So there is no noise, there is no cracks, there is no battle, there's nothing. It's just that those bacteria are caught by this IgA and they are kept at check. IgG also plays a role in maintaining the microbiome. But when IgG is going to get involved, there will be noise and there will be a battle, because IgG is one of those antibodies that is like a very strong police officer. When they show up, they have to show that they are strong, and they have to capture the bad guy, and they have to call in defense. So when IgG is involved in the microbiome, it will go ahead and activate the complement system. It will recruit neutrophils and macrophages. There will be an inflammatory process that will go on in order for those bacteria to go away. Why is that important? Because in healthy individuals, usually IgA is all what a healthy individual needs to keep the microbiome, bad microbes at bay. And the IgG is just sitting there, available if there is any problem, but in general does not do anything major. However, recent studies were done on individuals who have specific IgA deficiency, and many of those people actually are not even patients, and they don't have problems. And what we learned is, if you don't have IgA, then your IgG is going to come in and try to do a very good job. And usually in IgA deficient patients without any problem with anything else, it's going to keep things quiet for the most part also. So it's kind of really kind of seamless process. It will try to kick in. It's not going to do the exact great job that IgA does, but really a really good job that almost in most of the people, they're asymptomatic. However, the big problem happens when people do not have IgA or IgG, and that's the example of common variable immunodeficiency. And that's why I chose to use common variable immunodeficiency as a disease example, because this is one of the most common diseases in which affects adults, and we can do something about it. And we lack both IgA for the most part, and IgG is very, very low. So people who have CVID are dependent on their immunoglobulin replacement therapy to keep them healthy and prevent them from infections. But those immunoglobulins are coming from healthy individuals and not from patients. So it's like they have ability to bind bacteria, but they have ability to bind bacteria that is expected to be in the gut. And what we found in multiple, there was one study, beautiful study that came from Paris, that showed that the immunoglobulin products try to bind some of the bacteria in patients with CVID, but not as good as the IgG binds the bacteria in healthy individuals, because the bacteria in the gut of patients with CVID is different, as I'm going to show you. So I'm not going to go over studies in depth at all, But I want to highlight some of the key discoveries in the past decade and a half about CVID. This is perhaps the first times that we started thinking about the microbiome and CVID. And people started very early by just looking at the blood of patients with CVID, and they used to find some bioproducts of bacteria present in the blood of CVID patients, even when they are not sick. And more recently, in the work of Dr. Charlotte Cunningham Rundles from New York, she actually looked at the blood samples of a large number of CVID patients, and all of them were not sick at the time of the collection. She found that there is an increased number of circulating bacterial DNA in the blood of patients with CVID, particularly those who have CVID and some immune dysregulation, like platelet issues, lung issues, liver issues that we call non-infectious complications. So there is an increase of circulating DNA telling us that at some point, some of the bacteria that was in the gut was able to leak into the blood system and stay there with its fingerprint, which is DNA. There are several studies now that have looked at the stool samples of patients with CVID, and they have done with genetic sequencing. And if you recall, when we were talking about microbiome, we're talking about the genetics of those microbes, because this is really what we're studying. And they have found that the microbiome composition is different between CVID patients and healthy individuals. And if you were able to have patients who have CVID with immune complications, immune dysregulation, and those who only have infections, there was a difference. And if you compare those individuals to healthy controls, you'd see that there is a bigger difference between patients with CVID and healthy controls. If you look carefully at those studies, most of them did the sequencing that zooms in only to the level of the genus. So kind of like the big family tree, but cannot give you the individual person with their first and last name, if you will, which is the species of the bacteria. And they also, for the most part, have compared CVID patients to general healthy individuals. So it could be the healthy donor of the lab or the healthy donor of a different study. And we know that for the most part, the microbiome is impacted by diet and environment. So household members share more than 40% of the microbiome just for the fact that they live in the same household. Sometimes they share the same diet, and intimate couples share a very large portion of the microbiome because of a very close relationship. So what we wanted to contribute and is, knowing all what I've shared with you so far, our work that was supported with the IDF was trying to answer a couple of questions. First, what are those differences? Can we zoom in more deeply and understand in more details what are those differences between patients with CVID who only have infections and those who have immune dysregulation? And the bigger question that we will not be able to answer at 100%, but kind of trying to scratch the surface is, are those changes that we're seeing contributing to the immune dysregulation? And the last question that was not part of the research supported by the IDF, but our work that was supported by other work, we tried to look at the impact of the microbiome on germ-free mice. And I will not share the mouse part with you, but just as fast to tell you is that, you know, there are a lot of impact that the microbiome can do in humans, and we can study it in more clean way in mice, particularly in germ-free mice. What we did, and some of you might be contributors to this study, is we collected stool samples from our patients and their household controls. And we asked each individual to provide us 2 samples about 20 days apart. For the most part, we asked our patients to freeze their samples and ship it to us. And we did 16 sRNA sequencing, but we also went all the depths to the whole genome sequencing so we can tell the species, the first and the last name, and the exact individual of bacteria. And you'll hear something about diversity, which means richness. So the circle that has more slices is considered to be more diverse than the circle that has lower slices. And we looked also at the composition, which means who are those microbes? And this is using the genome sequencing. And I will tell you that we have found what others found that there are big differences between patients with CVID and immune dysregulation and between patients who have CVID and no immune dysregulation. And I'm not showing here in the slide, but when we compared between patients and their household controls, although we have really a small sample size, we were able to see that patients with CVID and immune dysregulation look different from their household controls, despite the fact that most of the household controls were spouses. They lived within the same house and they ate at least one meal together. So that tells you that this signature and difference in the gut microbiome composition in CVID patients and immune dysregulation is real. And even when you account for diet and environment, when you make sure that those are not different between the groups, you still see differences, which tells you that this difference is truly stemming from the disease itself. Our study added a little bit to the knowledge that we have, but collectively, we as a community, we have confirmed that there are disturbances in the gut microbiome, particularly in patients who have immune dysregulation. But there are a lot of questions that we don't know. Till now, we do not really know whether those microbiome changes that we're seeing in patients with immunodeficiency are causing the immune dysregulation or are a result from the immune deficiency. Because as I show you, there is a very intimate talk between the immune system and the microbes. Could it be that it's the immune system that is not functioning well that pushed the microbiome to be not as diverse? And perhaps it's both ways. But it's very important to try to answer this question, because if the microbiome changes are the cause for some of the immune dysregulation, then can we normalize the microbiome? And if we do, will that have a tangible impact on our patients? And for that, I'm going to mention to you some of the, and I'm going to move to just improve my light, I apologize. I'm going to mention to you some of the microbiome-based interventions that, that are currently being used. And I will just summarize very briefly what we know about it and how safe they are. I do need to mention that most of those interventions are done on healthy individuals or individuals with some GI disorders, but they are not particularly immune deficient, except in one situation that I'll mention when I mention it on. And very few of those interventions are FDA approved. But overall, if we take a very, very big picture of what do I mean by microbiome-based interventions? We are doing things to either introduce species to the microbiome. So we are ingesting things that have new bacteria inside our gut. Species depletion, which means that we are maybe taking medications to reduce or eliminate certain harmful microbes that are present in the gut. And enhanced growth. We are ingesting something to boost the growth of beneficial bacteria. Now, as I go through those approaches, you're going to see that one approach oftentimes tackles at least two of those three components, perhaps all of them. So they work together, but to kind of simplify things, we kind of try to think about it in a compartmentalized way. Perhaps you've heard about the fecal microbiota transplant, which is really an ancient intervention done by Chinese since the 4th century and used to refer to it as the yellow soup. And it's been went on and off through the medical interventions in life till it became now very popular, because you've probably heard that it's now FDA approved for prevention of C. diff colitis. The idea is simple. If you have an individual who's healthy and an individual who has significant GI symptoms because they are now engrafted by a lot of bad bacteria, why don't we take the good bacteria and give it to the individual who's sick and let this good bacteria eliminate the bad bacteria and live on? And in theory, this is very helpful and perhaps not very harmful, but the success rate in clinical trials has varied from 50% all the way to close to 97%. It really depends on how you give the microbiome. The most success that was seen is when you administer it through the rectum via colonoscopy or through enema. And it's more successful when you do this intervention multiple times. And that will lead to really change in the microbiome composition of the recipient. And the engraftment, which means will that good bacteria be able to take over the gut of the individual who's receiving it, really it varies depending on multiple factors. And there is this concept of a super donor, which means in clinical trials, they might have 10 or 20 or 30 donors, and they identify maybe 2 or 3 that all the people who receive the microbiome from those super donors engraft very well and have better results, which means that really there are specific bacteria or specific metabolites or specific set of factors that when they exist together, it works perfectly, but when they are not all together, the recipe is not complete and the outcome is not exactly what we expect. There is this concept also of autologous FMT, which was done in people with transplant. When we know that bone marrow transplant requires a lot of chemotherapy, you deplete the bacteria. So there were some clinical trials in which they collect the stool samples of the individuals themselves before they underwent an intervention that is very major. And then you give it back to them when they are undergoing this intervention. Let's say somebody's going to go on a very long course of antibiotics along with the chemotherapy, and it's found to be very helpful to prevent infections and febrile neutropenia and some transplants. But this is not an FDA approved. The only FDA approved treatment is this medicine here that is FDA approved to prevent recurrent C. diff colitis in individuals who are at high risk. It's only approved for adults for the time being, and those individuals are not acutely ill and they have already received antibiotics. But this is really a great first line of a starter. Now, anybody who knows my work about microbiome knows that I get a lot of questions about the diet and what should we eat to improve the microbiome. And of course, we have a lot of diets. And I will tell you that, yes, we favor healthy diets, but there have been no solid evidence to say that if you 100% change your diet, that you'll 100% get benefit. But definitely here in the picture, you see the Western diet that is high in processed food that we know has negative impact on the immune function and leads to bacterial overgrowth. You have the keto diet in which there is elimination of carbohydrates. You usually don't have a lot of elements that have a lot of fiber, but you focus more on high fat, high protein. It's helpful for people who have seizure disorder, but it has been shown that its impact on the gut microbiome is not 100% beneficial. So it might have enriched the gut microbiome with some non-beneficial bacteria over time. On the other side, you see that there is the whole plant-based diet that is enriched in fibers, and those fibers convert into short-chain fatty acids, which play a major role in immune system balance. So this is beneficial. And there is the Mediterranean diet that emphasizes plant food-based with olive oil, but there is protein in it also. And it has been shown to reduce the risk of multiple diseases like cancer, cardiovascular disease, but also promotes a healthy microbiome composition. And I'm going to zoom in on fermented food because I know that I get a lot of questions about that. And the idea is that fermented food has been made by really bacteria. For example, if we take yogurt as an example, the lactic acid bacteria has fermented the dairy into cheese or into yogurt. And when you consume that, you're consuming this live bacteria if you are consuming unheated yogurt and uncooked. And it's thought that those beneficial bacteria play a major role in our microbiome. Think about it, it's perhaps not the bacteria themselves because those bacteria do not live in our gut for a really long time, but it is the metabolites that those fermented foods are enriched with that perhaps promote a healthier microbiome composition and lead to long-term benefits. So for example, we recommend that people have fermented yogurt if they are going to go on a long course of antibiotics. You might have heard about prebiotics, and prebiotics do not contain bacteria per se, but they contain the fuel for the bacteria. So they have substrates like inulin or fructooligosaccharides or galactooligosaccharides, which really enhance the function of the bacteria to move towards a healthier metabolism, because as I mentioned earlier, the bacteria metabolize our nutrients. So people who have higher intake of fibers that contain a lot of prebiotics have usually a more diverse gut microbiome. And finally, probiotics, which are now available many times over the counter. And the idea is that this is kind of preserved live bacteria that has been shown to be helpful in preventing antibiotic-related diarrhea. Sometimes it's been helpful in irritable bowel disease, and there are even some studies in inflammatory bowel disease. You can either add it as an actual over-the-counter medication, but also, as I mentioned, yogurt and fermented food contain some of those probiotics because they contain live bacteria. So I know I took a little bit longer than what I was supposed to, but I wanted to kind of take the take-home message today is that patients with primary immunodeficiency in general and CVID in specific has been shown to have altered gut microbiome composition, but it remains unclear whether those changes in the gut microbiome are the cause or the result of the underlying immunodeficiency. And I really think it's a vicious cycle at the end of the day. The intervention benefits include enhancing the diet, and it might be helpful in patients with primary immunodeficiency. And there are further interventions like prebiotics, probiotics that might be helpful, but we have to be very mindful when we take measures that are very dramatic, because any of those interventions could have a downside to it on the way that you metabolize your medications, on the way that you absorb certain vitamins. And also, for example, for the fecal microbiome transplant, you cannot do it if somebody has a significant colitis, because you might end up with leaking of those bacteria into the bloodstream. So those interventions, although very promising, they are in their infancy, specifically for patients with immunodeficiency. We have to be very careful about using those interventions. The future is really evolving swiftly, and we wanted to move on to the next level. So we're really trying to go beyond just identifying the bacteria to understand more what's the function of those bacteria in the gut and their metabolites, because we believe that the microbiome is a metabolic organ and it functions in a very important way. But we know that the microbiome in people who live in the United States is going to be different from India, is going to be different from other parts of the world. And it's really not the exact bacteria that play a role, but the function of certain bacterial strain that is more important in driving the immune responses in our individuals. I'd like to end by thanking so many people who supported this work, including all my colleagues and mentors and students and technicians. A big thank you to the IDF. The IDF supported the human part of this research, which we are very, very grateful for, and we continue to use some of those funds in supporting the metabolic and metabolite work. And I would like to thank our patients and their families who contributed to this research. For sure, collecting samples for microbiome research is the least fun thing that you could do, but they were very graceful, and they have been always very helpful. And we are still welcoming samples for the metabolite studies. So if you are interested, feel free to reach out to the IDF or to me directly, and we're still accepting samples. And with that, I will stop and happy to take any questions.
Wonderful. Well, thank you so much, Dr. Hajjar. That was incredibly informative, and I know we have lots of folks who are eager to ask their questions. Again, my name is Emma Mertens. I'm the Program Manager of Community Health at IDF, and tonight's presenter is Dr. Hajjar from Texas Children's Hospital and Baylor College of Medicine. A friendly reminder that if you'd like to ask anything during the Q&A, to enter your question once only, please, in the box at the bottom of your Zoom screen. We have about, excuse me, 45 minutes left for questions, and we do have about 200 folks on the webinar tonight, so we will get through as many as we can in the next 45 minutes. We want to be mindful of Dr. Hajjar's time, but we ask that you please be patient with us, and we'll get through as many of these questions as we can. All right. So first question, we got a number of questions asking about the fecal microbiota transplant, which I think folks are really interested in hearing about. So just to kind of summarize all those questions in one, do you recommend or have experience with fecal microbiota transplant in people with PI?
We do have some experience, and also, as I might have mentioned, that some of the clinical trials that were done as part of the FDA approval included some patients with immunodeficiency. Many of them were secondary to medications or chemotherapy, but some of them had an actual primary immunodeficiency disorder. For the most part, in the clinical trials, the study was safe, and the patients were able to tolerate the FMT. It's very important to know that this is not an acute treatment, and the patients have to have met criteria, have been treated with antibiotics. The indication is not to treat an acute C. diff, but it's to prevent another C. diff from happening in people who have recurrent C. diff and are at high risk.
Thank you. All right, next question. This individual asks, is there a link between chronic sinusitis and gut microbiome?
That's a very interesting question. We have some people who looked at the lung and the nose flora, and they have some links between the nose microbiome and the lung microbiome and the recurrent infections in people who have recurrent infections with and without primary immunodeficiency. I will tell you that in the world of cancer, in which patients who have secondary immunodeficiency, the biggest link did come from the gut and did not come from the skin or the nasal cavity or the oral cavity. We don't have studies that directly link the gut microbiome with sinus infection, but I do think we can extrapolate from other studies that the gut microbiome is a major player to the systemic immune responses because it has the largest surface area interaction. So I would think that they are probably linked, but I cannot say that in an evidence-based manner because I did not come across any study that directly links the two.
Thank you, Dr. Hajjar. All right, next question. This individual says, I have atrophic gastritis. How does that affect the microbiome and CVID?
So the GI tract is very connected. And although most of what I've talked about in the gut microbiome is expected to reflect the lower part of the gastrointestinal tract, which is the colon, we know particularly in patients with CVID, this barrier between the different parts of the gastrointestinal tract is a little bit impaired. So we know that any inflammation at any part of the gut would literally affect the microbiome. Mindful that, generally speaking, the stomach is a sterile area in which there are no microbes unless there is an infection. So it is not normal for somebody to find microbiota in the gut unless somebody has H. pylori, and this is something that we notice, or somebody has an infection. But as I said, those barriers are not as strong in patients with immunodeficiency. So I believe that there is a link between any inflammation, any part of the gut, and the gut microbiome, but keeping in mind that generally speaking, the stomach is sterile unless there is an infection.
Thank you, Dr. Hajjar. All right, next question. Sorry about that, folks. All right, next question. How is a baby's gut affected when he or she is born and as they develop to a mother with primary immune deficiency, such as CVID, who already has an altered microbiome?
Right. That's a very important question. And the good news about that, our babies are very resilient, and maternal factors is one from many. So there are some data to suggest that exposure to antibiotics in utero could affect the baby's microbiome. And this is one of the major impacts. So if a mother with CVID had a lot of infections and was exposed to antibiotics, this potentially could impact the microbiome composition of the baby. But if the baby is born and they have their normal exposures and they have been fed and they are moving around, we believe that there is some resilience. So that slide that I showed with multiple factors, you really need to impact many of those factors together in order for you to impact the gut microbiome in children. So if it was just an exposure to antibiotics, but the babies are fed and they have their normal exposures, we don't know for sure, but we expect that, there's compensation mechanism.
Thank you. All right, next question. This individual would like to know, is it possible to improve the microbiome after decades of severe GI issues?
That's a great question. And I think this is the question that we're all trying to answer. I would say that many of the studies that were interventional to treat inflammatory bowel disease or colitis had mixed results. Particularly in CVID, the interventional study that tried to use a topical antibiotic that is ingested did not have positive results in CVID, but it was a small study and it was a short-term intervention. So I think this is really the million-dollar question. We definitely want to move towards generating a healthy microbiome in anybody, but I don't think we are there in a place and we can say that if you do A, B, C, then we expect that, that will reflect on your health.
Thank you, Dr. Hajjar. Next question. How can we improve our microbiome with probiotics and digestive enzymes?
So the probiotics are live bacteria, and they are expected to go through the stomach, stay alive, reach the colon, and do their job, which is metabolizing nutrients in a healthy way. So having probiotics in periods in which somebody is exposed to antibiotics is good. If they are traveling to parts of the world when there are risk of helminth infections, parasites, and things like that, it's good. I'm not sure what it's meant by enzymes. There are a lot of supplements over there, and my disclaimer is that I do not prescribe many of those supplements because I'm just, honestly speaking, not very familiar with what they entail. And I'm always a little bit careful about things that are not regulated by the FDA also.
Sure thing. Thank you so much. All right, I'm going to hand it over to Megan for the next couple questions, and then we'll transition back to me. So take it away, Megan.
Yes, thank you. So we've gotten a couple of different questions regarding dairy and the relationship between dairy and the microbiome. One person in particular is wondering who has an allergy to dairy, how that might affect their microbiome by not having any dairy intake.
Right. So, dairy falls into the fermented food because of those lactic acid bacteria. So I think there are a lot of alternatives in which there is fermented food that can contribute to the microbiome in a positive way. So and now even if you look, there are dairy-free yogurts that are present, and they are enriched with fermenting bacteria to try to compensate.
Thank you for that. We also have a question regarding IgA and IgM. If someone does not have either, is the gut microbiome in trouble from a stability standpoint?
If they have IgG, we usually see that the IgG steps in and compensates. If they don't have IgG and they are on immunoglobulin replacement therapy, I mentioned a little bit about it, that there was a study that shows that the IgG products try to step in, but they are not very helpful. But that's one aspect of it. But when we specifically looked at CVID patients, the one who have immune dysregulation were the mostly impaired in their gut microbiome composition. Many of the patients who have infections only also do not have IgM and IgA, but their microbiome resembled the healthy controls much better. So the antibodies contribute, but I don't think it's everything, because we're seeing differences within the CVID patients
Mhm.
themselves.
Thank you. And I'll hand it back to Emma for the next question.
Thanks, Megan. All right, next question. This individual, it's more of a comment than a question about the topic, but they were wondering, after this lecture, can you share the references of the papers you are citing and discussing? Would really love to read these papers.
I'll be happy to.
Awesome, thanks, Dr. Hajjar. All right, this next individual wants to know, have there been, this is kind of circling back to the FMT topic, have there been any studies looking at fecal transplants from healthy volunteers and patients with CVI and GI dysbiosis?
Yes, so some of the licensing studies that for that particular FDA-approved drug did have patients with immunodeficiency. I do not have access to the exact diagnoses of the immune deficient patients, but there were some patients who were immunodeficient, and those FMTs were coming from healthy donors.
Thank you. All right, we've also got a number of questions about diet. And this individual was wondering if you could provide some examples of prebiotic foods.
Yes, so I have some in the presentation, and they are in things like garlic. They are in some of the whole grains as well. They are some of the kimchi and some of the fermented food are also expected to have some prebiotics.
Thank you. And I just want to add to that when we upload the video of this webinar, we'll also include Dr. Hajjar's slides, so you'll be able to see some of those examples in there. All right, next question. Have you seen any research on the use of a very low carb or carnivore diet to improve the microbiome?
So I'm not sure if low carb diet, they're referring to the keto diet, because that's kind of what comes to my mind. So the keto diet is low carb, high in protein and high in fat. And although it's beneficial for weight loss and seizures, I think the lack of fibers that come from the lack of a lot of the fruits and vegetables have actually, in a way, negatively impacted the microbiome in the studies that looked at it.
Thank you, Dr. Hajjar. All right, next question. Do you advise someone who has been on the low FODMAP diet for long term, meaning they've been on this diet for years, if they have persistent intestinal bacterial overgrowth? So I think they're asking, should I continue on the low FODMAP diet?
I'm not sure I caught the name of the diet. Low what?
Low FODMAP. I
have to say I'm not familiar with that diet. I'm sorry.
We can move to the next one then. All right, this individual wants to know if someone has a severe reaction to prebiotics like inulin and other fibers and has CVID, is there anything else they can do to increase the good bacteria in their microbiome?
Right. So there have been some studies that show that not everybody will respond equally to high fiber diet, and that highly depends on the microbiome composition. So there are certain bacteria that digest the fibers better than others. So and there were some studies that looked at that, particularly in the weight loss. So sometimes the microbiome composition is not ready to take on this high fiber diet. And I think that, you know, from the bottom of the things, there are some things that we can do to impact the microbiome, but there are many things that we can't because of the immune system and maybe genetic susceptibility. But I would say always that having a balanced diet in which you're not tipping off the balance toward one thing way more than the other is good. Fermented foods provide the prebiotic component and provide the good bacteria at the same time. So it might be something that people can lean towards if they are not able to ingest a lot of the prebiotic.
you, Dr. Hajjar. All right, next question. This individual asks, is there any good consumer test that allows one to keep track of the diversity of his or her microbiome? If so, what would that test be, and how accurate is it giving the complexity of the human microbiome?
Right. So I think that goes to the idea of personalized medicine that, we kind of understand what is there in the individual's gut microbiome and tailor our therapies towards this. And this might happen in the future, but I would say that in the world of microbiome, when you sequence every individual patient, the amount of data that comes out is huge, and the amount of variability, even from day to day, is significant. So that's why where we are right now requires a lot of people to participate in a single study in order for you to see any differences, because the data are not individualized at this time, but rather you're looking at a signal that resonates in many individuals. So I do not think we are yet at a place in which you would go and sequence the individual's fecal matter and say, okay, this is your diversity, this is what we need to do. Perhaps in the future, and particularly if we have more information about the metabolites and the function, then that might become possible. But at this time, it's just going to be so much messy data that it's just not going to be meaningful on an individual level.
Thanks, Dr. Hajjar. All right, next question. This is kind of a two-part question. This individual asks, is the gut microbiome connected to the microbiome of the skin? If so, how are they connected? And are CVID patients more likely to experience colonization on the skin due to their microbiome?
That's a very interesting question. I am not aware of studies that looked at CVID particularly, but I am a collaborator on a study in cancer patients in which they have looked at their stool samples and they have looked at their skin microbiome also. And it really depends on what disease you're studying. So you're going to find a signal, and there are expected bacteria to be existing on the skin, and there are expected bacteria that is supposed to exist in the gut. And most of the studies are trying to see if there are changes in either or that can predict disease or side effects of medications. And I mentioned that earlier, that when particularly asking a question about response to cancer immunotherapy, it seems that the strongest signal come from the gut and not from the skin.
Thank you. All right, next question, getting back to FMT. This individual wants to know, has there been any research on norovirus and fecal transplant? CVID patients cannot clear norovirus because of their poor gut microbiome, especially those with immune dysregulation.
That's a wonderful question, and we have tried to look at that. And so anecdotally, if a patient has norovirus and C. diff colitis and they receive FMT for the C. diff colitis, oftentimes the norovirus gets better as well. But the FMT is not FDA approved for norovirus, so it is not possible to treat people with FMT for norovirus specifically. The anecdotes happen when they have C. diff on top, and when you treat with FMT, a lot of times they do respond.
Thank you. We've gotten a couple questions about, I think it's SIBO, S-I-B-O, so I'm going to ask those next. What can be done for PI patients about SIBO or leaky gut?
So just having all the measures that I mentioned that promote a healthy microbiome is good. If there is a particular evidence of bacterial overgrowth or a leaky gut in which there is evidence of systemic infection or significant diarrhea, then there are times in which we use certain kind of antibiotics to see if that helps. And we usually work with the GI doctors quite a bit on those particular cases. But it has to be very individualized.
Yeah. All right. And then I wanted to circle back to the questions just while we're on the topic of SIBO about the low FODMAP diet. So Megan did a little research to see if we could kind of figure out what that was so we could answer this person's question. Low FODMAP diet is used to treat and prevent SIBO or IBS symptoms. FODMAP stands for fermentable oligosaccharides, disaccharides, monosaccharides, and polyols. So I think what you were talking about, I know you mentioned fermented foods like kimchi or like, what's another fermented food? I know you mentioned them in your talk.
Even the dairy is considered to be fermented. And
yes. So that individual was asking if they've been on that type of diet. Let's see. If they've been on that type of diet, so focusing on sort of those fermented foods for long term, would you advise continuing on that type of diet if they have persistent intestinal bacterial overgrowth?
If the change in the diet had made a difference in the outcome, I think it's helpful and not harmful. So I would say if it's helping, for sure, to continue.
Thank you. And thanks for circling back with me on that one. I wanted to try and answer that question since that person took the time to put it in. So thanks, Dr. Hajjar.
Absolutely. Thank you for looking it up.
Yeah. All right. Next question. What does sugar do to your microbiome?
So sugar is a substrate like all the other substrates, and it gets taken up by the bacteria. I don't remember seeing a lot of research, particularly on sugar, so I'm not going to claim that I know, except that, for example, if you don't have the enzymes that are supposed to break a certain kind of sugar, then you might end up having more bloating. For example, we know a lot about lactose insufficiency, in which you cannot digest this particular sugar, so you get diarrhea and bloating and lactose intolerance.
Thank you. Next question. Does coffee consumption affect a CVID patient's microbiome?
I have not come across caffeine's impact, so I really don't know.
Right. This individual wants to know, does the health of your microbiome affect your mental health?
So I did not touch upon, there is a large body of research that looks at the gut-brain axis, and certainly there are a lot of data that suggests that there is a direct link, because the bioproducts of the bacteria, there is a leaky gut and can circulate in the blood and actually cross the blood-brain barrier and impact the brain functionality. It's beyond the scope of my expertise, except that I have come across those data several times in the past, and the body of evidence is increasing.
Thank you. All right, this next question is sort of a chicken or the egg scenario. What's your best guess as to which came first, CVID or altered gut microbiome?
You know, I think it's the question that we're truly trying to understand, and I don't think we have. I think there is a susceptibility that is inherent in patients who are destined to have CVID. And that susceptibility, does it come from the fact that they had recurrent infections and been on antibiotics and disrupted their microbiome early on before they were even diagnosed, knowing that CVID takes a long time between the time people start having symptoms and between the time of diagnosis, or they have exposures in their environment that led them to changes in the microbiome, and that led to significant immune dysregulation. I really think it probably two hits. So one hit is that a person was destined to have CVID. And the second hit is the number of environmental factors that gave a second hit to the microbiome. And I think it's more of like a threshold. So if you meet that threshold, then you have this hit from a genetic or whatever genetic pool that we think drives the CVID process. and another hit on number of factors that affected the microbiome, you tip the balance, and now you reach that no return spot, and now you have a disrupted microbiome and an immunodeficiency that will lead to immune dysregulation.
Thank you, Dr. Hajjar. All right, next question. This individual wants to know, is Crohn's disease, and I'll say an IBS and also ulcerative colitis, just kind of lumping those all together, linked to PI?
Certainly, there are a lot of immunodeficiency disorders in which inflammatory bowel disease are part of the phenotype. So oftentimes Crohn's and ulcerative colitis symptoms appear in some of our patients. We call it colitis, but sometimes on pathology it's not exactly the same, and that's why I say Now, in very young children, there is something called very early onset IBD, inflammatory bowel disease. And depending on how young the children who are less than 6 years old, many times they have very early onset IBD on the background of an underlying immunodeficiency disorder.
Thank you. All right, next question. So Dr. Hajjar, during your talk, you mentioned how folks who live together or are in the same family or are partners might have similar microbiomes due to the environment, eating similar diets, being in a similar environment, that type of thing. So this individual wants to know, I'm curious as to how having an immune deficient spouse, themselves having CVID, and an immune suppressed husband, their husband had a double lung transplant, could affect maintaining healthy microbiomes for each individual.
That's very interesting. So the studies oftentimes were done on healthy individuals, and they show that there are a lot of similarities between individuals who live in the same household. There are even some studies that were done on people and their pets, and they found that people and their pets, particularly dogs, can share a good percentage of the microbiome. If both individuals have a dysbiosis, meaning that an imbalanced microbiome composition, they might share it also in the same token. But most of the studies were looking at certain kind of most of the studies that were done in the context of healthy individuals, and I don't remember coming across a study that particularly looked at a negative impact, like if one of the people had a disease that is associated with dysbiosis, how would the impact? Now, obviously, there are infections that can be highly contagious within the same household, like C. diff, which is typically something that is highly contagious. But if we're talking about a steady state in which there is dysbiosis with no active infection or diarrhea, I'm not sure I know the answer to this question.
Thanks, Dr. Hajjar. All right, circling back to fecal microbiota transplant. We're getting tons of questions about that. I think folks are really interested. So this individual wanted to circle back to the one question that was asked about FMT and norovirus. And they are asking, when norovirus has cleared with C. diff, has there been follow-up to see if immune dysregulation numbers such as CD8 have improved?
I do not think we have this data. No.
Okay. All right, next question. Is there any known connection between gastroparesis and the microbiome?
I mean, hypothetically, yes, because the kinetics and the movements of the gut is dependent on the nerves, but also dependent on the microbiome composition. But I don't think I have researched that particular area specifically, because it's beyond the scope of my research, which is way more about immune interactions rather than the motility of the gut.
Certainly. All right, and we are getting close on time. I definitely want to be mindful of your schedule, Dr. Hajjar. So I think this is going to be our last question of the evening. This individual wants to know, is a person's microbiome the same across all of their organs? So across gut, skin, lung, etc., or does each individual organ have its own microbiome?
Yes, I think this is a very important question, and the answer is it's different. So there are certain microbes that we know exist in the oral cavity. There are certain microbes that we know are normal to be found in the stool. There are certain bacteria that are known to be found on the skin and so on. We do see that there are some oral bacteria that is present in more abundance in the gut in patients with inflammatory bowel disease or immunodeficiency because of that barrier integrity impairment that I mentioned about earlier. That in healthy individuals, this is very compartmentalized, and because each of the bacteria provides a different function. So you should be able to sequence and kind of guess that this is coming from the skin versus from the oral cavity versus from the stool and so on.
Thank you so much, Dr. Hajjar. That is going to wrap up our Q&A for this evening. I can't tell you how much we appreciate you taking the time to answer everyone's questions. And thank you so much to our audience for your participation and engagement. These were some really excellent questions. We love hearing from our community, and we really appreciate you sending in your questions for Dr. Hajjar. Thank you so much, Dr. Hajjar. And I know it's after work hours, so if you need to run, we totally understand. I have just a couple more slides about upcoming events, and then we'll close it out for this evening.
Well, thank you so much for having me, and thank you for all the great questions that you have a tough crowd. I could not answer many of them, but that I appreciate you and Megan for your help as well. Have a great evening.
Thank you, Dr. Hajjar. Take care.
Bye-bye.
All right, again, thank you everyone for taking the time to enter your questions this evening, and a big thank you to Dr. Hajjar for her time. If your question was not addressed during the program, go online and submit your question to Ask IDF. Visit www.primaryimmune.org/ask-IDF or give us a call at that 800 number on your screen. We have a wonderful resource navigator who will personally connect with you to answer your question or direct you to appropriate resources. And you can also check out the IDF Resource Center, support services, and YouTube page to find more videos and resources. It's a busy fall here at IDF, and we hope to see you at one of our many upcoming programs. Our next webinar will be on October 18th, covering PI and lung issues. And we also have 2 lunch and learns coming up in October, one covering XLA and the other talking about ADA-SCID gene therapy updates. We are also hosting 2 in-person education days in Chicago and Anaheim. Visit www.primaryimmune.org/events to register and learn more. Finally, we hope you will save the date for our 2024 PI conference, which we are thrilled to share will be hosted in person at the Sheridan Grand Riverwalk, Chicago from June 20th to 22nd. Thank you so much again to our wonderful sponsors for their generous support of IDF's educational programming and community services. We truly could not offer these types of programs without their support. Thank you everyone again for your participation this evening. We really appreciate all of your wonderful, tough questions for Dr. Hajjar. And we thank Dr. Hajjar again for her time and her expertise on this very important topic for our community. We hope to see you at an IDF event this fall. Take care and have a great rest of your evening.